Okay, thank you very much. So my name is Takafumi Ueno from Tokyo Tech, the Department of Life Science and Technology. So first of all, I greatly appreciate the Nakamura Sensei and the organizing committee for this opportunity to attend the kind of the interdisciplinary environment and the international environment. Because it's the symposium, this first time to attend the ELSI the program for me. But this is very impressive because previously I worked in Kyoto University, Enzymes. That is the first generation of the WPI. That's why I love the kind of the environment. But when we coming to the Tokyo Tech 8 years ago, I miss the kind of the interdisciplinary field environment in my department because my department isolated in the Suzukage campus. Anyway, so this is just a joke for the local people. Today, let us start my real scientific presentations. Right. So they say that today my presentation title, Artificial Enzymes Using a Protein Assembly. We're gonna interested in control and functionalize the chemically about the protein assembly because previously many researchers, the chemists, interested in the modification and functionalization of protein, but they are focusing monomer, like a small molecule. But right now we are not focusing if we learn more about the more huge assembly structure of the protein as the building block the small compound chemicals, so we can expand the chemical techniques to the nanoscience and biology. That's why so we are interested in studying, investigating about this target of the protein assembly. Before moving today the main topic, so let me introduce my research background briefly by using the several slides. At first, look at the schematic slide of the systematical diagram of the protein and the peptide to the living cell. The several small elements that can be combined to obtain the amino acid related as a building block of the peptide. Peptide can, of course, easily converted to the protein in our biological system. After the different types of the protein or homogeneous protein can be integrated to form the protein assembly with various functions like an enzyme and storage and transduction and so on. After that, so these different types of protein, when they are integrated, so we obtain organ. Then finally, we obtain cell. In this slide, please pay attention the size of each component. When I think about the small size called as a nanoscale, then this is a microscale. It's the real scientific world of biology. The protein assembly is located in the intermediate size between the nanoscale and the microscale, with a scale between the 10 to 100 nanometer. That's why, so when we chemists want to modify and synthesize, understand the mechanism of the protein assembly are it too large. On the other hand, the biologist can want to the biological reaction and structure mechanism in detail that's too small, right? That's why, so the mesoscale molecule like in a protein assembly, the field of this research, it's like missing parts between the chemistry and the biology. So our motivation is that's why, so other motivation, if we can establish the new scientific field by using in the field of the mesoscale molecule, so we can, of course, expand the chemistry and science between current nanoscience and biological science. But of course, as you know very well, when we want to use a natural protein assembly, like a photosynthesis tool or something, so these are too complicated to manipulate chemically. That's why right now we can use only the very, very simple protein assembly. So I show here the several very typical the protein assembly, which in which our laboratory member using now. So this is the natural protein cage, which is the one of the most famous protein assembly, which is can serve as the iron storage in our biological system. Then this is a protein needle. It's a very unique artificial assembly structure getting from the bacteriophage T4. And the other one is also very unique. That is, of course, the one with the protein assembly is obtained from protein crystal. So today, a little bit to introduce briefly about the previous our research progress by using the protein cage. They move to today's main topic by using protein crystal. This is a very unique. The original the function is the iron storage. That's why so we can easily concentrate and accumulate different types of the metal complex or metal sources within this protein cage instead of the iron or iron oxide. This idea is bringing us to the very interesting functional chemical functionalization. So if we introduce the metal, sorry, I need So instead of the accumulation of the iron cation, so if we can successfully introduce the metal complex catalyst or metal nanoparticle, right? So we can promote the chemical reaction and the catalytic reaction within the nano caged environment constructed by the protein assembly cage. So this based on this concept. Thank you. Too late. Anyway, so this is the previously more than 10 years ago. So we had first established the preparation of nanoparticle, which usually they utilize for the organic synthesis in organic solvent. So this the heterogeneous catalyst can easily promoted within the protein cage, and by the incorporation of organometallic catalyst or especially, so we can successfully obtain the polymer, synthetic polymer within the cage. And of course, we can combine the different types of metal complex catalyst within the protein cage. We can proceed the cascade reaction, but this cascade reaction just like in the proof of the concept, not so high efficiency compared with the previous slide, okay, previous lecture. So this means the advantage to use a protein cage. So we can utilize this original biocompatibility. That's why so we can easily deliver this system into the living cell, and we can promote the bio-orthogonal catalytic reaction by using this protein cage. So, sorry. So this is we are previously established this kind of the functionalization of the protein cage by using the incorporation of the metal source or metal complex catalyst. But still now, this is a serious problem. Is hard to apply to any kind of the field, because of course, as you know very well, the protein when we manipulate chemically protein, so the serious problem is the stability and the low yield compared with the molecular catalyst or inorganic catalyst. The wizard to overcoming this problem, so we it is very, very hard to develop for the further application. So but of course we know that the very unique potential of the protein assembly as a molecular scaffold to promote any kind of the catalytic reaction. So that time, so I was looking for the new types of the protein assembly scaffold instead of the protein cage. Then we found that the protein crystal as the next generation of protein scaffold and molecular scaffold for the catalytic reaction. Then maybe you know very well about the protein crystal. The protein crystal is prepared to understand the molecular structure of the protein. Because when we use the protein crystal as the source of the diffraction, then after the radiation X-ray, we can get a lot of diffraction to determine the crystal structure of the protein. But in order to the protein crystal, in order to synthesize the protein crystal, so it's many researchers reported many kind of the method to obtain this, right? Then and analyze this original physical property. So as shown here, so that is the most important things. It's the density of the protein in protein crystal. It's about 300 milligram per ml. It's quite same to that of the inside cell. So this means if the the precisely packed, if the protein precisely packed within the protein crystal, so this is the existing in the same environment, similar environment in the living cell. This, in other words, so this means the protein can activate like within the living cell. And additionally, so the stability, with respect to the stability of the protein itself, so protein is located in the protein crystal. So protein shows us that the highest stability compared with in aggregation or in solution. That's why without using the protein crystal, we never expand more about our chemistry. That's why so we eager to use the functionalize the protein crystal. And additionally, so the protein crystal as shown here, the protein crystal, you can easily image the monomer structure of the protein. But you please keep in the remember, so this protein crystal structure came from the protein crystal. The protein crystal made from the precisely assembly structure of the protein. That's why so when we see the protein crystal as an assembly structure like this protein crystal have the very unique internal porous structure, like the typical porous material like zeolite or mesoporous silica and MAF. So I show here the very unique property, porous property. These porous property are comparable to the data those of the typical porous material. That's why so we can modify the porous protein crystal like zeolite or MAF or other typical porous crystal or porous material. We can expand and combine the catalytic activity of the protein inside crystal to any kind of application. So, but unfortunately, so the very less number of the report related to the application. In my knowledge, so this is the first application, first report, describing about the protein crystal as novel microporous material, and around the 2000. Because of course, you know, this protein crystal shows us that very great potential as this molecular scaffold, but still very difficult to obtain the protein crystal, right? We want to our ideal protein crystal. At first, we have to prepare the protein, right, with the extremely purified level, and then crystallize. That we don't know how long to getting the crystal, right? And after that, we have to analyze the crystal structure to show the kind of the very beautiful porous structure. That's why, so this is very, very terrible things at around 2000, right? But recently, so several researchers involved in our research group, so developed the different types of the functionalization of the crystal, by the immobilization, different types of the material. And moreover, so recently, so the protein crystal one of the target of the modern protein engineering, so de novo design of the protein, right? And of course, we investigated the different types of the functionalization method by using the protein crystal like that, right? So, but still, from this method and the publication, we have not overcame the serious inherent problem of the crystallization, about crystallization to obtain the larger amount of this material as the molecular scaffold for the catalytic reaction. So today, I will talk about the current research progress related to the protein crystal as the molecular scaffold for the catalytic design. The first one is the basic part of my motivation of this research project, the preparation of solid artificial metal enzyme. The second one is the enzyme encapsulated within the in-cell crystal, protein crystal. Then finally, I just introduce our latest the unpublished results, the designing the cascade reaction in a protein crystal. The first move to the first topic. So in the case of the protein crystallization and the crystal structure analysis, many researchers are interested in structure of the metal complex bind coordinated to the protein, because the protein and metal complex can serve as their meta drug. That's why several researchers are interested in coordination, interaction, and the structure that attaching to the surface of the protein. That's why several researchers reported the metal complex and coordination structure of metal complex. This is a very simple example by using the lifetime crystal, because the lifetime crystal it's easily handled, easily obtained after the crystallization for the overnight. So we can easily obtain that with the high resolution quality of the crystal. Then from the previous research, so we know that if we introduce a metal complex catalyst instead of the kind of the typical organometal complexes, so we can prepare the solid heterogeneous artificial metal enzyme. But of course, the problem was the instability, less stability of the protein crystal itself of the lifetime. So in order to overcome this problem of the less stability of the original protein crystal, so we use the very typical technical cross-linking treatment. When the crystal dipped into the solution containing the cross-linker like glutaraldehyde, this is the strongly cross-link the protein crystal within several days. After that, so this protein crystal cannot never be dissolved in a buffer solution or pure water or organic solvent. After the treatment of this cross-linking reagent, so this cross-linking lifetime crystal dipped into the typical mixture solvent containing the ruthenium metal complex catalyst with 30% DMSO. So even after this treatment, this crystal still retaining with the original morphology, right? Then this is finally we analyzed the element. We did the elemental analysis. The ruthenium bind to the protein. About one ruthenium complex bind to the one lysine molecule. After the treatment and the purification, washing the excess metal complex, we try to use this composite for the catalyst like this, for this hydrogenation reaction in the buffer solution, right? So I show here the comparison of this metal crystal composite, and this is a composite dissolved in solution, and only the original metal complex catalyst source. This is the protein crystal. It shows us that the highest time conversion efficiency with the enantiomer activity. But when we use the soluble complex as a catalyst, they did show the very less effectivity for the catalytic activity. That's why the crystal itself, it's a very robust scaffold for the catalytic reaction, even in the water solution, right? The next advantage to use the lifetime crystal. Lifetime crystal, when we use the crystallite with a different condition, so lifetime crystal show us that the different lattice structure, the different space group. So when we use this tetragonal crystal as a catalytic scaffold, so especially the larger bulk when we use the bulk substrate, so this is show us that the conversion and this kind of the enantiomer activity R isomer, S isomer. But when we deposit the metal complex catalyst with a different orthorhombic crystal of the lifetime, so the same reaction proceeded, but the enantiomer activity completely opposite the isomer obtained from with the protein crystal of the lifetime. So this means this is in order to understand the mechanism. So we discussed about the crystal structure comparison as shown here. So in both cases, the ruthenium complex, tightly bound to the histidine 1,5 with the same coordination structure. But the channel location and the channel morphology quite different to each other, right? That's why when the substrate access to the bind catalytic center, so the direction should be different in both crystal. So this is one of the reason why the orthogonal crystal and the tetragonal crystal proceed this catalytic reaction with opposite the enantiomer selectivity, right? So this is one of the demonstration to construct the metal heterogeneous artificial metal enzyme. This is applicable for the different types of the construction of the heterogeneous artificial metal enzyme. That when we incorporated the dimolecule and the precursor of the metal cation within this. After that, so we prepare the platinum nanoparticle within the protein crystal by the light ionization or sodium borohydride reduction. Then after they obtain this composite containing the nanoparticle of platinum, right? So this is show us that the high performance for the hydrogen evolution. So this is one of the different techniques to construct the heterogeneous artificial metal enzyme. This is bring us the stability, original stability of the close-linked protein crystal that bring us the kind of the catalytic design by using the biomolecule scaffold, right? So, but still now, even by using the scaffold, still the inherent problem remaining. That is the very, very hard task, hard work to purify the protein and crystallization with a large scale. I never ask my students because this business is too much for the current students, right? That's why. So instead of the kind of preparation by our service, we want to look for the other system to obtain the large amount of the protein crystal. We found one possibility that this means that I show here. So usually, when we use the user cell to get the protein, we thought that only the purification of the protein dissolve in the solution. But several living cell, spontaneously prepare the crystal after production of the protein, as shown here. For example, oxidase enzyme in peroxisome and this is IgG or TBCAT-B can be spontaneously crystallized within the living cell after the synthesis of them within the living cell. If we can use this kind of system to obtain our desired protein crystal as a scaffold of the catalytic reaction, so we can expand. We doesn't care about the large-scale synthesis of the molecular scaffold of the protein crystal. Then, on this reason, we are using the in vivo crystallization. Then, especially we are interested in the very unique system of the protein crystallization in the living cell of the cytoplasmic polyhydrolysis virus, CPV. When the CPV is a very famous insect virus. This CPV, when the CPV infected to the insect cell, so insect cell synthesize the polyhydrolyl protein, crystalline polyhydrolyl protein, as well as the preparation, the replication of virus itself. During this life cycle, so after the production of the virus spontaneously encapsulated the polyhedral crystal. The polyhedral crystallized in the cytoplasm. Then finally, so several large number of the virus can be encapsulated within the crystal. The crystal, the role is the protector armor of this virus for virus to survive the longer time, about several years, against the harsh condition, dry condition, the cold condition, the hot condition. After that, so go again to the midgut of the insect. So this is the dissolve in the mid gas solution around the pH 10. Then again, start the life cycle of the CPV virus nanoparticle. Then this is still this crystal structure, the reported microarbitrator of the crystal structure of the polyhedral. So this is the when we got this crystal structure, so we can design precisely the chemical with our chemical sense. Then this means our purpose is to the chemical and the protein engineering of this crystal. So the original wild type, the dissolve in the lower pH 10. But if we dissolve this crystal at the optimal pH, and instead of the encapsulation of the virus, we can encapsulate our target enzyme. So we can prepare this crystalline composite, one-pot synthesis within a single cell, right? So this is we successfully obtained this kind of the composite. But the serious problem was this original design, how we can melt the crystal to get the protein or enzyme still retaining the original enzymatic activity. In order to do that, again, go back to the molecular design based on the crystal structure. So of course, the protein, each protein, fixed with the crystal with the intermolecular interaction with neighboring molecule. So this we analyze the neighboring molecule interaction like that. We are focused on the N-terminal H1 helix. The H1 helix exposed to the other neighboring molecule. Then this is the retaining the kind of the multiple hydrogen bonding network with others. So if we reduce the hydrogen bonding network, so I expected that this crystal should be dissolved easily at lower pH compared with pH 10. Then we replace the arginine 30 to the lysine or alanine. Then we obtain the crystal successfully. Then we at first, in order to incorporation of protein, we incorporate the GFP with the H1 tag, right? Then the method have already reported microarbitrator. Then we can actually confirm the crystallization and the encapsulation of the GFP by using the mutant like this. Then we evaluated that the dissolving efficiency of the mutant. Mutant then can easily release the GFP around the pH 8 to 8.5. They compare with the higher pH, the dissolve and the GFP released from the wild type around the pH 9 to pH 9.5. That's why, so at first, our chemical design, protein engineering, successfully utilized for this dissolving the behavior, right? So next, instead of the GFP, we incorporated PKC, which is the most and one of the typical phosphorylated enzyme. So this is incorporated within the mutant wild type. After that, so this is the treatment dry condition for 1 week, because this crystal can survive the dry condition. And after the 1 week, dissolved again the buffer solution to release the enzyme incorporated within the crystal. Then we evaluate the enzymatic activity with a commercial screening kit. I show here the results. Before the drying, so the mutant and the wild type show us that the efficient enzymatic activity, as well as the free enzyme. But after drying for 1 week, so the free enzyme is completely denatured. That's why it cannot show the original enzymatic activity. But after the 1 week, even then after 1 week, so the protein crystals surviving, and the enzyme, retaining the original enzymatic activity within the crystal. That's why, so second. Sorry. So this is one of the demonstration of the encapsulation. But as a final goal, we want to use the protein crystal as and we want to promote the chemical reaction with the protein crystal. That's why, so this is by applying this original method for the combination of the encapsulation of the two types of the enzyme like this. Then in order to do that, so we designed more about the molecular scaffold, the polyhedral crystal. Then by the deletion of the 3 amino acid residue, 38 amino acid residue to enlarge internal space. By using this, they successfully obtain the kind of the crystal encapsulating the two kind of the enzymes like this. Then I show here the confirm the same amount of the enzyme of the lipase and the ADH, the encapsulated in the wild type and the mutants. Then next, we evaluated the cascade reaction by using this substrate. So this substrate at first the hydrolyzed by the lipase. After that, so they eliminated with the ADH. Then finally, we obtained this product to confirm by using the confocal microscope. That first, we confirm this cascade reaction by as shown here. That when we use the wild type as a scaffold to encapsulate the two kind of the lipase and ADH, the wild type composite didn't show the high intensity the fluorescent confirmed by the confocal microscope. But when we use the delta 38 the amino acid deleted mutants, so this mutant composite shows us that more higher intensity as shown here of the product assigned to the product as here. And next, I show here the comparison of cascade activity. That first, we are compared with the mixture of each protein crystal composite containing the only lipase and ADH, right? This is also the mixture. So this also can promote this cascade reaction in the solution. But the cascade reaction is a very lower the catalytic activity compared with the dual enzyme encapsulated material. Then moreover, so this among compared among all the mutants and composite. So delta 38 amino acid deleted mutants show us that the highest catalytic efficiency. So I show here a little bit discuss about this reason. So when we use the mixture of this lipase composite and
we have only limited time for
sorry.
Q&A.
Sorry. Anyway, so I want to say that we are successfully obtained the efficiency protein crystal composite. Then finally, so this is one of the next generation of the protein engineering by using protein crystal. That's why so we are investigating more more efficient, effective catalytic performance by using this system. But finally, thank you very much, all of you.
Thank you very much. So now's the question time. So we want to take questions from the audience. Okay. Ladies first. Ladies first.
Thank you for your talk. I really enjoyed it. I have a question to the first or second part. I mean, you make a protein crystal and added some metals to put a catalytic center. So I want to ask you about the stability of the crystal machinery, because I guess that the crystallized condition and the conditions that used for catalytic reaction is very different. And if I understand correctly, not only the metal side, but the surrounding protein part also works for the substrate selectivity or something.
Yeah, of course,
so
right.
I'm wondering the stability of the crystallized
Yes,
structure.
right. So in the case of that is a very, very important part for the designing this kind of the composite. So enforce the stability of the protein crystal by using the cross-linked treatment, cross-linking. That's why, so after the soaking and the deposition of metal complex by using the organic solvent, after that, the completely remove the excess metal complex, then dry completely under vacuum. But after coming back to the buffer solution, we can get diffraction from the crystal. when we use the lifetime crystal. Of course, we will try to different types of the crystal from the different protein. Never get the diffraction data. Right. That's why depending on first important things, which the protein it utilized, which protein crystal it utilized as a scaffold. Right. Thank you.
Yeah. I'm going to ask about the cascade that you wanted to show at the very end. Can you talk about how the intermediate is transported between the sites? Is it diffusing through the crystal? Is that diffusion fast? Does it have a different diffusion coefficient than your reactants and products?
Yes.
Those could actually affect the outcome of the reaction. Do you know anything about that?
Yeah. That's a very important point as well. But we don't know the analyze. Right.
Okay.
That's why I need your help.
Okay, fine. Does it change the kinetics? Okay,
Yeah.
we'll get to that. Yeah,
Yes.
it's an important thing.
And actually, it's a little bit hard to determine the diffusion coefficients of the intermediate product and the substrate within the crystal.
Yeah.
Right now. Right. So we think about this.
Okay. Thank you.
Right. Analysis method. Right. Thank you.
So we also have one question from Slido. So you listed the sequence, the order of the stability in your slide, and so white crystal is more stable than aggregation and then in solution. So where do liquid crystals belong in this series in terms of structure stability?
I don't know the liquid crystal. That's why. So yeah, I just using the solid crystal.
Okay.
Right.
Actually, I have a question. Sorry. So this is the final talk. So can I take additional one question? So you mentioned you achieved some chiral selection in your protein crystal.
Yeah.
And actually, this is very important for prebiotic synthesis. We would like to selectively generate L amino acid from scratch. So I wonder, how is it possible to use a polypeptide instead of a natural protein to realize the similar function?
Yeah, in the case of when we consider about the protein crystal, right, they're a little bit difficult to crystallize the small peptide because small peptide have the high flexibility, hard to crystallize, right? Of course, several researchers want to prepare the kind of biopolymer catalyst, but still they're available in solution. But the terrible thing is hard to understand the mechanism because without a little bit difficult to determine the crystal structure of the composite. That's why. So if we overcome this part, so we can apply any kind of synthetic short peptide as a scaffold for that kind of the crystal
Okay, I see.
material.
Thank you. Thank you. So thank you all. So this is the end of this session, and hope you enjoyed this session. And now we have coffee break.